Good day, this is Professor drwillphd, PhD. And today's Tuesday, July 12th, 2022. It is 6:37 and 8 seconds, a.m. Eastern Standard Time. Today's research, cancer cells. So let's hop to it here. The programmed cell death in cancer cells, overcoming resistance to apoptosis, inducing compounds. Date July 11, 2022. Source, Tokyo University of Science. Induced programmed cell death, such as apoptosis, is a widely used therapeutic option for the treatment of cancer. Unfortunately, many cancer cells become resistant to programmed cell death and continue multiplying. In a new study, researchers have synthesized new complex hybrid compounds named tryptosine peptide hybrids, which successfully induce a kind of programmed cell death known as apoptosis in Jurkat cells, a type of lymphocytes. And these apoptosis-inducing compounds may revolutionize cancer therapy in the future. Other than apoptosis, several types of programmed cell death occur in cells, including apoptosis, necroptosis, and autophagy. And of these, apoptosis is the most recently identified type of programmed cell death, which is caused by the influx of excess calcium in the cells, leading to cell death. The cancer cells often become resistant to drugs that induce apoptosis and other types of programmed cell death. In such cases, inducing apoptosis, which is not dependent on caspases, can act as a promising anti-cancer treatment. Hence, the development of compounds that can induce apoptosis in cancer cells is crucial. Researchers conducted a study to develop novel, complex peptide hybrids with apoptosis-inducing potential. And previously, researchers synthesized an iridium complex peptide hybrid compound and observed that it induced cell death in cancer cells, which was different from apoptosis. And since this compound was unlike other apoptosis-inducing compounds, researchers wanted to understand its mechanism and apoptosis induction. The goal now is to synthesize new compounds and elucidate how they induce apoptosis in cells. The newly synthesized compounds were composed of a tryptosine core, an aromatic hydrocarbon with 2 or 3 cationic peptides made up of amino acids, lysine and glycine. The team subsequently performed experiments on Jurkat cells, a type of immortalized human lymphocytes used in research to understand the type of programmed cell death that occurred in these cells on treatment with SYN6 and NT6. They found that death in these cells was inhibited by carbonyl cyanide, M-chlorophenylhydrazone, which is an upcoupling reagent and inhibitor of mitochondrial calcium uptake. However, cell death was not inhibited by inhibitors of other types of programmed cell death, and hence they ruled out autophagy, necroptosis, and apoptosis, confirming that apoptosis is a major programmed cell death pathway induced by SYN6 and NT6 in Jurkat cells. The studies have indicated that the TPHs, SYN6 and NT6, induce the transfer of excess calcium from the endoplasmic reticulum to mitochondria, resulting in a loss of mitochondrial membrane potential. It is very likely that these phenomena are strongly related with the fusion of endoplasmic reticulum with the mitochondria, followed by cytoplasmic vacuolization, resulting in cell death. When asked why these 2 TPHs were selected for the study, the TPHs, SYN6 and NT6, are more hydrophilic than other TPHs, which can also be a reason for their high apoptosis, inducing anti-cancer potential. And through additional imaging experiments, the team detected the presence of cytoplasmic vacuolization, elevated mitochondrial calcium concentrations, and the degradation of endoplasmic reticulum in Jurkat cells treated with SYN6 and NT6. Based on previous findings, the team hypothesized that in Jurkat cells as well, the influx of calcium The mitochondria might be facilitated by the close proximity of the endoplasmic reticulum and the mitochondria. As expected, they found that the endoplasmic reticulum and the mitochondrial membranes were attached to one another, facilitating direct transfer of calcium. And these findings confirm that Jurkat cells treated with Sin-6 and Anti-6 had undergone programmed cell death owing to paraapoptosis. They also provide crucial information for the design of compounds that can be used as therapeutic agents against cancer and other diseases. Good day, this is Professor Will, CBMD PhD, Professor of Microbiology, Bioinformatics, and Pathophysiology. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.